This method can help answer key questions in the bone metastasis research field, such as what changes are induced in the bone marrow adipocyte growth and development in a cancer microenvironment. The main advantages of this technique are that it is easy to perform and that the candidate molecules can be quickly obtained.
This method can provide insight into the crosstalk between bone marrow adipocytes and melanoma cells. It can also be applied to other cell types such as osteoblasts.
Begin by seeding 2 × 10⁵ B16 F10 melanoma cells in a 100 mm dish with 10 ml of DMEM supplemented with 10% FBS for a 24-hour incubation at 37 degrees Celsius and 5% CO2. The next day, wash the about 80% confluent cell culture 2 times with 5 ml of PBS per wash, and starve the cells with 10 ml of serum-free culture medium in the cell culture incubator for 18 to 24 hours. The next day, use a pipette to transfer the conditioned supernatant into a 50 ml conical tube for centrifugation, and load the medium into a syringe equipped with a 0.45 micron pore filter. Then strain the solution into a sterile container to remove any remaining cell debris and store the conditioned medium at -20 degrees Celsius for up to 1 month. To induce bone marrow adipocytes, culture mouse F14 1.1 bone